All right, guys, welcome to day two, week two, day two of this lab. So what we're going to be doing is we're going to be taking and doing our counts for our bacteria. And so what we're going to start with is we're going to start with our first bacteria plate, which was the comparison between before and after hand sanitizer or hand washing, and that was up to you. And so your plates probably look something similar to mine. There's a bunch of stuff growing on it, and you're probably looking at it saying like, okay, how am I supposed to count like these giant blobs that are going on in here? And the answer to that is you're going to guesstimate. So there's no exact. I'm going to borrow my son's Lego, his little Lego knife to do some pointing. So there's no like great way to count like these big blobby areas. But if you look closely, you might see, and let me see, I'll try not to get the reflection then in there. If you look close, you might be able to make out and distinguish individual colonies even within the large blob. And what you'll see is you'll see they're bordered like circles. So there'll be like little circles within the big blob. Now, I'm not going to count that big blob as anything other than just maybe like one or two colonies. What I'm gonna do is I'm gonna count. There's like tiny little there. You'll see them on yours. There's tiny little pin-sized colonies. They're like little dots that make up that big blob. I am gonna count those. And so there's two parts that we're going to be counting. We're going to be counting the diversity, so that's the different types of bacteria. And then we're also going to be counting the number of colonies. Now, the number of colonies is going to take a long time because there's a lot of them. So I'm not going to record while I'm doing that. What I'm going to do is I'm going to count them up and I'm going to record them on this sheet over here. I'm going to show you where you can record them. So you have your lab document right here. You have your lab document here. And so what you're able to do is you can just literally punch in the number of colonies here before and after and all the different types of bacteria, and you can add those in there. And then let me show you one more. So this is what I'm going to show you guys is, let me do something. So I'm going to show you this. So we're going to be doing, I'm going to show you how to make graphs in Excel. And so you guys can definitely do this by hand. You can definitely use the graphs that are provided in the lab that I gave you. So you can print them out and then do them by hand. No problem. But for those of you that want to learn Excel, and I know that there's somebody out there that does, because I talked to your wife and she said you're excited to learn this. So we are going to do this in Excel. So what I'm going to do is I'm going to count up the number of colonies. That's the number of individual dots, right? And they can be big circles or they can be little circles on my before side. And I'm going to enter that in there where that green box is. And then I'm going to count up the number of little circles on the after side. And then I'm going to count the different types of bacteria. And you might look at this and say, okay, how am I supposed to distinguish types? Like how am I supposed to distinguish types? Well, there are different colors and different types of colonies within here. Now, what you'll maybe be able to see is that some of them kind of have a light gray hue, and then some of them are a little bit lighter, kind of almost a white hue. And so to me, I don't see much more than maybe, and then sometimes you may or may not, you may see like yellowish ones, and those would be a different type of bacteria. And so it's your job just to count up the number of dots on the before side, record that data, count up the number of dots or colonies on the after side, record that data, and then look to see how many different types. And for me, there's only going to be maybe 2 or 3 tops on either side. So I'm going to let you do that. I'm going to pause the recording and then you do the same, count them up, and then I'll go over how to make the graph. Okay, so I counted mine up. Now, it's tricky to count, right? Because you're probably freaking out, like, how am I going to be able to identify all of these and make sure that I'm not accidentally recounting them? The answer is you don't. Like, you're just going to have to do the best you can. So, like, don't overthink it. Don't stress out about this. Try to get as close to an accurate representation as you can, but don't sweat it if you're not there, or if it's not perfect. Okay, so what I'm gonna do is I'm gonna share my screen again. So I'm gonna come over here to Excel, and what I found, if my computer cooperates, is I found there was probably about 150, 140 colonies on my before side, and maybe a few less than that, maybe almost 100 on my after side. Now, I did notice that there was Your typical kind of big blobby white ones, and then your little freckly white ones. And then I noticed there was a reddish colored one and a yellow colored colony and on my before side. And so I count that as 4 different colonies or 4 different types. On my after side, all I saw was the dots and then kind of the blobbish ones. And so I just counted that as 2. Now what we need to do is we need to make 2 different graphs. We need to make one graph for the number of colonies and one graph for diversity. So if you want to take a second and set up your Excel spreadsheet just like mine, it's really simple. I'll show you how to do it. So you would just type in like number of colonies in one cell, and then the next cell over, you could type in diversity. And what's nice is if you come up here to the top between the K and the L and you double click on the line, it'll actually make your cells expand so that, so you don't have the words running out of the cell. And then all we're going to do is write in before over here, after down below it. And then I just took a box like this and I came up here to this, border box selection menu, and I selected all borders, and then boom, there you have it. So you can type your data in like that. That's what I did over here. Okay. So what I'm going to do is I'm going to select these colonies, or this quadrant. Now I'm not including diversity because I'm not graphing diversity. I'm just graphing before and after number of colonies. So I have my x-axis variable, my y-axis. The number of colonies is the dependent variable because it's dependent on which side I sampled from. Okay, so next I'm going to come up here and I'm going to take my data and I'm going to go into, go back. I'm going to go to insert. Okay, I'm going to move my guy down. I'm going to insert, and I'm going to come over here, and I see there's this like kind of chart looking thing up here. So we got 2D column. That looks pretty good to me. So if you come across into the middle, it should pop up like a little dialog box that tells you what that graph is, and it's a column or a bar chart. That's perfect. So we're just going to click the first one. That one should be fine. And then boom, I got my graph. Oops. So I'm going to hit Control Z. I didn't mean to move that. And what I want to do is there's a couple of things I do need to add to this graph before I turn it in. The first is the X and Y labels. Now, granted, this is going to work out just fine. I also need to change the title of the graph. So this would be hand washing effectiveness on bacteria. Okay, so I got hand washing effectiveness on bacteria. I just try to center this up a little bit, make it look pretty. Okay, now I still need to add in my X and Y axis variable. So I'm going to come up here to the upper left hand corner and I'm going to add a chart element, and you'll see it has axes. So I got like I can take away my axis labels, or I can go to axis titles and I can give myself a horizontal and I can give myself a vertical. So I'm going to do both. So the x-axis is before, this is my independent variable, before or after hand washing. Okay. Give that a second to update. Sorry, my computer's running really slow. I think it's because I have a bunch of programs running. All right, and then we're going to come up here and we're going to add the other axis label, which is the vertical one. Okay, and then I just double click on that, and then I enter this in. This is the number of bacterial colonies present. There you go. And it's that simple. I mean, it's not pretty yet. You can move the elements around a little bit, but ultimately this is the desired figure that I would present based on my data. And so that's how you make a graph of the number of colonies in diversity, or just of the number of colonies. Now we need to do diversity. So I'm going to select my before and after. This is a little bit tricky, right? So what you can do is hold down button control, and then it helps you select multiple cells. So I'm going to select control, and I selected all these cells. So I selected before, after, diversity, and then 4 and 2. Now I'm going to come back up here to insert. I'm going to come over to my bar graphs, click on 2D column, And oh my gosh, what is happening? Everything looks weird, right? It says I've got all these series and stuff. So we're gonna have to troubleshoot this, which is good because we should probably troubleshoot stuff like this together. So what's good is that it's showing me 2 versus 4, right, for my diversity. The problem is it's telling me there's all these series. So what I need to do is I need to go up to my select data. So I need to figure out where the data is coming from for this. And what I need to do is I need to remove a lot of these series. So I'm just going to click remove. Oh, what did I do? I want series 5. So let's see, horizontal axis label. I'm going to come over here and I'm going to change my horizontal axis label, edit to right here. Boom. Okay, so before and after. Then I accidentally, what did I do? Yeah, I'm going to start over again. Okay, so we messed something up. Let's figure this out. Select data. All right, I definitely do not want series 1 or series 2 or series, oh, I need that one. All right, hold on. I'm gonna pause this while I figure this out so I don't waste your time. Okay, I figured out what I did. It was super easy. Okay, so I selected diversity. It is one of those categories. So what we need to do is click on before, hold down control, click after, so that selects both of them, and then just click 4 and 2. That's it. You don't have to click on the diversity. That's why it got all messed up. So go to insert, and then we'll go up here to our 2D bar graph, and then look, bam, there it is, right? And that looks perfect. So I'm gonna move this out of the way. I'm gonna move this over, and then we gotta change the chart title. So this is hand washing on bacteria diversity. Hand washing on bacteria diversity. And then the only thing we really need to add to this are just our axis labels. So I'm going to come up here to the upper left hand corner, add chart element, axis titles, primary horizontal. This is before or after hand washing, as if that wasn't obvious. You still need to label it. And then add a chart element again, axis title, vertical. And then we're going to add in diversity of bacteria. This is number of types. Yeah. So there you have it. So we've got a title for our chart. I'm actually going to put these next to each other real quick. Let's kind of like shrink them up a little bit. There we go. Shrink them up a little bit and then put them next to each other. So there you go. So that's how you do hand washing effectiveness on bacteria, hand washing on diversity, and then that's how you put in your labels. So hopefully that answers that question for you. Okay, so let's go back. I'm going to unshare my screen if I can. I'm going to unshare this screen, and then we're going to go back, and what we're going to do next is count our body parts. And so I want you, if you're going to do this in your, if you're going to do it by hand on paper, that's great. Go ahead. If you're going to do it in Excel, maybe set up something like I have here, where you have 6 different categories of body parts, and then you'll count the number of colonies and the diversity, and then go ahead and do that. So what you're going to notice is that you actually have separate Petri dishes for all of this. Let me see if I can get back out of here. What you're going to notice is that you have a Petri dish for, you know, like that's my inanimate object one, but you should have 3 Petri dishes all from different body parts. So I've got fridge handle, front door. That's an inanimate object. So you've got different body parts. You've got behind the ear, armpit, that kind of stuff. And then let's see. That's an inanimate object. Before and after. All right, so you should, yep. So I've got all my body parts here. Oh, there's my last one. Oh yeah, belly button, side of nose. Let's see it better like that. Belly button, side of nose. And so take your 3 Petri dishes that you have divided up, and that's because they're all divided in half, you should have 6 things that you're counting. And so I'm going to share this one more time. Those counts are all going to go into Excel over here. So you're going to be able to write in the body part, write the number of colonies, and then diversity. Okay, so go ahead and do that. We'll pause the video and then we'll come back. Okay. I almost forgot to add. So if it makes it easier for you guys, you can use the magnifying glass that's in your lab kit to help you do your bacterial counts. Just a side. Okay, so that took me a couple of minutes to do all these counts. And I put them into our table over here that I'm sharing with you guys. You can add them into your own table. Notice that my numbers are not specific. They're like 400, 225. So one of the things that I do that kind of like makes it easier is I'll count like a third of the plate, or maybe half of the plate, or maybe like 25% of the plate, and then I'll extrapolate. So I'm like, oh, if I counted 140 on a quarter of the plate, I'll multiply that by 4 to give me the whole plate. So it saves a lot of time. If your stuff looks like super decorated, like my lower back and upper thigh did, I just looked at this and I was like, okay, there's upwards of thousands on there. I'm not going to be able to count thousands of these. So what I did was I just said, oh, you know what? There's more than 500 on those places. And so what's really important though is that we get to the graphing part. And so what I'm going to do is I'm going to select the data set for the number of colonies. And I'm just going to select belly button all the way down to upper thigh and just the number of colonies for this one. So then we're going to go up to our insert and then we'll click on our bar chart. And let's see how we're doing here. Maybe grab everything. So body part and number of colonies. Let's see if that works. Well, it doesn't look like it's wanting. Oh, I know why. Because I put over 500. That's what's thrown it off. So when I put more than 500, that's why it's like it's not an actual data set or data point, and so it's like freaking out about that. That's what's happening. Okay, so even though it's more than 500, right? I'm just gonna say 500, and I'm gonna select all my data set. I'm gonna come up here, and then see how easy that was. Boom. Okay. All right, so I have number of colonies. Number of colonies is actually my that's my y-axis label, and then the body part should be my x-axis label. So my chart title can be comparison of bacterial presence on body parts. Yeah, there you go. And then I'll take this chart and I'll say, okay, come up to my add chart element, and I'm going to go axis title, primary horizontal, and that's where I'm going to type in the body part. So body part is on the x-axis, and then I'm going to click on this chart again, come up to my add chart element, axis titles vertical, double click on that, and then that's going to be my number of colonies. So there we go. And so that's how I do that. Go ahead and let's do diversity together. So I'm going to select belly button all the way down to upper thigh. So you can click here and then hold the shift button down, and that'll select all those. And now hit the control button down and click here, 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 here. Okay, so now I have all of these selected with all of the diversities. So I'm going to come up to insert. I'm going to go bar chart. It's not working. I'm not sure why. Let's make sure that we have our categories down. So let's select our data set. So let's see. We want to edit that. We want the series name to be all of these. We want the series values to be all of these. There we go. Okay. So in case you're wondering what I just did some magic there. So let's cancel that. So let's pretend like you guys probably got something similar to me. Oops, I don't want to move that. Control Z. I just want to move the whole chart. So this chart looks all jacked up. We're like, wait a second. That's not showing me what I want. So I'm going to come up here to my select data, because now I can manually tell it where the data is. So I select data. Now this whole like data set is belly button, side of nose. That's fine because that's the series, right? That's the different x-axis labels. But now I need to tell it, I need to edit it and tell it where to get its data from. So I'm going to click on edit. So the series name is what do I name all of the different bars that I'm going to produce, like all of these bars. And so I click on that. And then the values, that's going to be the data range. And so I click on that arrow. Oop. See how it says series values? And then I'm going to select all of these. So, and then hit OK. Hit OK. Now the problem here, the only problem is that my series, my 1, 2, 3, 4, 5, and 6, they don't have labels. So let's go ahead and go back up here to select data. And maybe it's just that I need to get the labels, right? So it says horizontal category axis labels. Oh, well, that's easy. So hit edit, axis label. So I'm just going to come over here and select all of these. Boom. And then it should populate it with belly button, behind your armpit, lower back, upper thigh. So one of the things we haven't done yet is provided x-axis label and then a y-axis label. So we've got our chart. Let's come up to add chart element, axis titles, primary horizontal. And what goes there? Body part. So body part is the independent variable from which we were sampling. The dependent variable is going to be the y-axis. So I've clicked on just kind of the side of my chart here, and I'm going to come up to add chart elements, axis titles, primary vertical. Click on that, and I'm double clicking here, and this is going to be number, or I should say diversity of types of bacteria. There we go. And so what I found was on my body, I didn't notice there was a whole lot of difference between the diversity. There was definitely differences in numbers where I sampled. That made a big difference. But as far as like the diversity, I didn't see a big difference. So that was interesting. So next we're going to do our inanimate objects. And if you decided to do the extra credit from the other video, you can also include that in your inanimate object chart or whatever you'd like, or you can make your own chart for that. Now, I'm going to let you guys, I'm going to stop sharing the screen, and we're going to go back to our, we're going to go back to Google Chrome. So I'm going to share this screen with you. So let's go back to Google Chrome. All right, so we've done this in Excel. So remember, if you've done it in Excel, you can copy and paste your graphs over and put them into your Word document and upload that. That's totally fine. You can definitely answer, right, your conclusions. So where were we at? This was for hand washing. So you're going to have a conclusion, right? Total number of colonies, number of diversity of types of colonies. You're going to have a conclusion. Was your conclusion about the number of bacteria supported? Was your prediction about the diversity of bacteria after hand washing, was that supported? So you can look back at your chart and see if that was supported or not. Okay, and then we did it for body parts, so we're going to skip that. We already populated this table in Excel. You should have done that already. We've made our 2 graphs in Excel, so we're done with that. And then we did inanimate objects, and so now I want you guys on your own, right, from what we've learned, I want you to be able to count up your inanimate objects and make some graphs, and you can do it here if you'd like on your paper by hand, or you can do it in Excel. And then finally, we've got some questions that we're going to answer. And so I just want to run through those really quick with you guys. So you are given 2 slides by a scientist, told one slide contains an organism in the domain bacteria, and the slide and another slide an organism in the domain eukarya, but he forgot to tell you which slide was which. So there's 2 ways you could recognize these. All right. The first way is that eukaryotic organisms, which have nucleus and organelles and things like that, they're going to be way bigger. The bacteria are going to be way smaller. I mean, bacteria are like 1/10 of the size of a eukaryotic cell on average. So they're very different in size. Another way that you could tell is organelles, the presence of organelles. Bacteria don't have organelles, but eukaryotic organisms do. So if you see organelles inside the cell, that's a good example. So that's 2 ways. So now you guys are going to finish this off. Which of your body parts exhibited the most bacteria number? Why do you think that was? Which had the most diversity? And you could say maybe the diversity wasn't that great. And my hypothesis is that my skin everywhere is going to have it's going to have different numbers of bacteria, but it's going to have the same types of bacteria. It's almost like a microbiome that covers your body. And then you can answer the questions about the inanimate objects. So as a general pattern, did you find more on your body parts or did you find more on the inanimate objects? And then you can create just a hypothesis. You don't have to test it about why that's the case. And so all you need to do at this point is you just need to take a picture of you and your bacterial plates. That's going to be one photo. So just show me that you did the experiment. And then you're going to submit a completed copy of this lab to Canvas. So you can either do it electronically, like in a docx, like a format, or you can take your PDF making software if you wrote it out by hand. So you got two, sorry, I'm rambling, but you have two different things you can do. You can either do it digitally and upload all your photos into your, like you can take the Excel graphs that we did and put them into an electronic Google Doc or a docx format and then upload that to Canvas. Or if you printed it out and you did your graphs by hand, you just take a picture of that. So you'll have a picture of yourself with your bacterial dishes, and then you'll have either an electronic submission or you'll have like the PDF where you, or the JPEG of you taking photos of your actual work. And that is it for our lab. So I hope this was beneficial to you guys. I hope you learned something with Excel. Hopefully I didn't expose how not good I am at the new version of Excel. It seems like Excel puts out these new versions and they're more user friendly, but they're different than the old versions. And so it's a little bit tricky for those of us that are used to like the old versions to work on the new ones. So good stuff though. It was good for me to go back and kind of figure that stuff out. So, all right, you guys, have a good one. We'll see you.